Hi everyone, welcome to my channel Pure Biology, and today I come with another topic, that is the myosin. Okay, and in my previous video I explained about the actin filament, and today I will try to explain about the myosin. Okay, before going to this topic, I want to say thank you to all of you who have already subscribed my channel, and you also support me a lot for the growth of this channel, and that's why I am very thankful towards you. Okay, and Merry Christmas, but belated Merry Christmas to all of my viewers, and obviously Happy New Year in advance. Okay, all you just remain happily with your family. Okay, so this is the topic myosin. What is myosin? Myosin is actin filament based motor protein. Okay, this is the actin filament based motor protein, and the form of the myosin that is found in muscle. Okay, and generally most important myosin, that is the myosin 2 molecule. Okay, so what is saying that the myosin, that is the one kind of actin filament based motor protein, and generally we can see myosin 2 in different type of cells, especially in the muscle, and another myosin also present, that's like 1, 2, 1, 3, 4 and 14, etc. These are all found in the eukaryotic cell. Okay, and this myosin molecule, myosin 2 molecule, is an oligomer. This is an oligomer, and this is composed of one pair of identical heavy chains and two pairs of non-identical light chains. Clear? Two pairs of non-identical light chains and one pair of heavy chains, and each of heavy chains that has a globular head. Okay, globular head domains, and where this domain remain? This remain at its N-terminus region. Okay, this is present in the N-terminus or N-terminal region, and it contains actin binding site. Okay, because this is the region where the actin filament they can bind with the myosin, and obviously this is the catalytic site. Why this catalytic site is needed? This is needed for the hydrolysis of ATP. Okay, each myosin head that binds and hydrolyzes ATP, how? It using the energy of ATP, and in that way it work towards the plus end of an actin filament. So again I repeat this part, that myosin contains two pair of non-identical light chains and one pair of heavy chains, and heavy chains that contain the globular head domains. Okay, and these contain an actin binding site and catalytic site, both the region or both the sites present at the where this is the N-terminal region, and this region only that where the actin filament can bind with the myosin, and obviously the hydrolysis of ATP, this is also occur here where the catalytic site. Okay, and by this work, this myosin can work towards the plus end of an actin filament. So what help here? This hydrolysis process or hydrolyzing process of ATP that helps the myosin to move towards the plus end of the actin filament for binding with this actin filament. Clear? And the globular head domain, that is followed by a very long amino acid sequence. So simple word is that globular head domain that contains the amino acid chain or amino acid sequences that forms an extended left-handed coil tail This is the brief diagram or rough diagram, you can say. These are the tail region, okay, and this is the head region. And the two light chains bind close to the N-terminal head domain. Okay, so where they can bind? They generally closely bind at the N-terminal head domain. Okay, a major role. Okay, one thing is that this demineralization is another important thing that mediates. So which one mediates? This extended left-handed coil tail. That means this coil tail. This coil tail needs help in the demineralization process. Okay, and the rod-like tail of the myosin 2. This myosin 2, the rod-like tail of this myosin 2 is generally allow the molecules to polymerize into bipolar filaments. Okay, to polymerize into bipolar filaments. Okay. Okay, so what is saying that the demineralization? No, demineralization is not. This bipolar filaments. The rod-like tail of the myosin 2 is to allow the molecules to polymerization into bipolar filaments. So rod-like tail that helps in the polymerization into the bipolar filaments. And the long coil tail that bundles itself with the tails of other myosin. Its another role is it can try to bind with the other myosin molecule. Okay. So generally the head region and the tail region. So head region is that containing the globular head domain, and that is containing the amino acid sequences, and the tail, that is the coiling tail, it mediates the demineralization. And another important function is that also this long coil tail, this bundles itself with the tail of other myosin molecules. And this tail interaction result in the formation of the large bipolar thick filament. So one filament when binds with the other filament, in that way a thick filament of myosin 2 molecule or myosin molecule that is formed. Clear? And that have in that way several hundred myosin heads oriented in opposite directions. Okay, so in that way several hundred of myosin heads or myosin filament, you can say, that form. And when the myosin 1 molecule that is treated, myosin 1 molecules not means that myosin 2 is not myosin 1 2. No, myosin 2 molecules but single filament. It is treated with the trypsin protein, then it is clipped into two fragments. One is the light meromycin and another is the heavy meromycin. Clear? And the light meromycin that consisting of the alpha helical rod, and heavy meromycin that consists of the globular heads. The subsequent treatment of the heavy meromycin with the papain that release the two globular protein heads, and called one is the S1 subfragment and another is called the S2 subfragment. So the common so what is saying here, if one single filament of myosin is treated with the trypsin, then divides into two forms. One is the head region containing portion and another is the tail region containing portion. Then again treated with the papain, then what will happen? Then the two heads S1 and S2, that means this region again clipped and form one S2, the two head-like structure you can see that are called the S2 subfragment, and this form the S1 subfragment. Okay. So the common feature of the all myosin is that motor domain or the motor head must be present, and uh, generally the other domain, also sometimes other domain present, but their function vary from myosin to myosin. So mainly motor domain is common and that is present in all kinds of myosin, but another domain may be present for another function. Okay, but common feature is that every myosin that has a motor domain. Clear? So this is the structure of the myosin I have explained here, and this is a very important part. One is the actin filament and another is the myosin. Those two are very important, and where it is very important? In muscle contraction, because if myosin and actin filament both are not present, so the muscle contraction is not possible. So what is the role of this actin myosin? We will learn about in the process of muscle contraction. We will learn, okay, and I will try to explain in my next video. So continue with my channel, and thank you.